everyone. My name is Jianping Fu. I'm a full professor at the Department of Mechanical Engineering Department at the University of Michigan, Ann Arbor. So welcome to the Integrated Biosystems and Biomechanics Laboratory.
Hi, my name is Aiden. I'm going to be showing you around the lab facilities. So here we share a lab with other top researchers in the field of small-scale biomechanics. Our lab specializes in human development and embryology using microfluidic tools to make stem cell models. So we combine our biological knowledge and our engineering training to not only ask daring questions to move the field forward, but also to develop groundbreaking new microfluidic tools to answer those questions. Our lab is located here in this building, G.G. Brown. G.G. Brown is the mechanical engineering building on the north end of the University of Michigan's Ann Arbor campus. Ann Arbor is located near Detroit in southeast Michigan, surrounded by the Great Lakes of North America.
Hi, my name is Sajida. So we are using PDMS to generate microfluidic devices in our lab. And here is the station that we are preparing PDMS to control the mechanical properties of the PDMS. So we are preparing PDMS here, and we have some modes for the microfluidics that we need to pour them with PDMS.
So welcome to our cell culturing room. This is where we perform our cell culturing in these hoods so that we can add synthetic extracellular matrix, signaling molecules, and cells to our microfluidic devices so that we can control the exposure of cell colonies to chemical or mechanical stimuli. So here we have a video microscope which helps us to obtain quality images to help us document our progress. And here we can see we are using a special microfluidic device to generate these cell colonies that will form into embryonic-like sacs, and we can expose them to certain drugs to examine how they might affect early post-implantation development.
Hello, my name is Qifeng, and in our lab, this is where we start to obtain our data. So this machine is a real-time PCR machine, and using this machine, we can check the gene expression in our cells. In this place is the immunostaining station. Here we can add fluorescent antibodies to target certain proteins, and then using the fluorescent microscope, we can check the expression and localization of these proteins. By doing this, we will know whether our cells are behaving properly or are these differentiating into a certain cell types.
Hey everyone, my name is Robin. Welcome to our fluorescent microscopy room. Here we have access to two wide-field active fluorescent microscopes and one spinning disc confocal microscope in the next room. We use those systems to examine the fluorescent markers inside our cell colonies, allowing us to see what's going on in those tissues. So besides our own microscope, we also have access to the microscopic core, one of the core research facilities here at the University of Michigan. So here we are at the Norway nanofabrication facility. This is a clean room where we use photolithography and deep reactive ion etch to make the wafers for our microfluidic devices. So in addition to L1F, we also have access to core facilities that can help us conduct gene editing, lentiviral transfection, as well as single-cell RNA sequencing.
Microtest is something really special because I'm reading all of these papers in microfluidics, and the fact that I have the opportunity to meet and listen to the top researchers in this field who are publishing all those papers is an incredibly exciting opportunity.
Microtest is one of the best conferences in the microfluidic field. I'm very excited to present my work and talk to the people, talk to the experts from all over the world.
Thank you for joining us on this virtual tour of the lab, and we look forward to seeing you in this year's µTAS conference.
Welcome to the Microfluidics and Biological Physics Lab. We use microfluidics to develop diagnostic devices, particularly with a focus to affordable healthcare. But we also use microfluidics to address biological questions that cannot be addressed in other experimental systems. So let me show around the lab area. Okay? So as we enter the lab, Over this side, we have our fabrication area. We have a plasma bonder, a spray coater, a solid ink printer for paper fluidic devices, and one of the two 3D printers that we have in our lab. This is the area where we do all the molecular biology work, and this is where we cut and punch our PDMS chips. And over here, we have, this is where we do mostly the electronics and mechanical kind of work. We have a press for hot embossing, and this is the area where we handle bacteria and blood samples. So entry is restricted over there. And now finally, this room over here is the microscopy room. So this is where we do microscopy and microfluidics as well.
Hi, I'm Shiljitin. This is the room where we do our microfluidics and microscopy work. We have a Nikon fluorescence microscope as well as a line of portable digital microscope developed by our lab members.
Hi, I'm Saurav. This is the compact nanoimaging tool where we can imprint nanoscale features in our microfluidic devices either by using thermal imprint or UV imprint lithography.
Hi, I'm John. This is the clean room of IIT Bombay's nanofabrication facility, which we extensively use for photolithography. Hi, I am Ramesh. I studied how bacteria move, grow and divide in microfluidic containers. All my experiments are performed in this shared FPD-based confocal microscope.
Hi, I'm Sarita. I measure the stiffness of single healthy and diseased fabrices in a high throughput manner using a microfluidic device.
Hi, I am developing a foam-free microfluidic step emulsification device for the generation of droplets and gel beads for the sensitive detection of pathogens.
Hi, I am Somdatta. I have developed an isothermal amplification protocol in droplets for the detection of malarial parasite.
Hi, I have developed this centrifugal microfluidic platform that can handle large sample volume without using syringe pumps. Now I am using this platform for detection of circulating tumors.
Hi again. Here I am trying to capture cancer cells on the basis of their size and deformability using a radial pillar device.
I'm Kamal Prakash Nair. I'm a PhD scholar here, and my work is on surface acoustic wave devices. And currently I'm working on using these SAW devices for acoustic manipulation of cells, and more specifically the separation of blood plasma from blood for diagnostic applications, and also on the portability and process time aspects.
Hi, I'm Aksha. I work with Russell Microfluidics for isolation of exosomes and circulating tumor cells from biological tissues.
Hello, Amtaji.
Hi, I'm Gauri.
Watch out for our Qasaimeh µTAS, one microfluidic universal gradient generator that can develop gradients of different functional forms by simply changing the fluorides.
We use a positive pressure pump and a homegrown microscope to conduct our experiments.
Hello, I am Santosh, administrative assistant of this lab. I handle all the logistics.
We look forward to µTAS as it gives us a platform to present our work, and we will get to network and learn from experts and peers in the field.
µTAS, being one of the largest conferences in the world for device miniaturization and microfluidics, will be a great stage to showcase my work and also get valuable feedback from like-minded individuals. But more importantly, I'll also get to see the cutting-edge research from people from all around the world, and especially in the field of interdisciplinary sciences.
So our group has been attending µTAS since 2015, and we try to be there every year. It's a very exciting conference, gives us a chance to see a wide range of activities in microfluidics and gives us networking opportunities. Even though we attended it virtually last year and will be attending virtually this year as well, I really look forward to interacting with everybody. So see you at µTAS.
Hello everyone, I'm Mohammad Qasaimeh from New York University, Abu Dhabi. Welcome to our lab tour. Let's get started.
Hello everyone, this is Mohammad Deliorman. Let me show you the lab space we have.
My name is Sam Shofela, and this is just part of the tour. I'm giving you an overview of our soft lithography workbench and where we assemble all our microfluidic devices. And we have the lab right here, where we have the workbench, the mixers, and all other devices we use for soft lithography.
Hello, my name is Ayub Glia, and let me show you where we are located. So our lab is located in this building here, which is part of NYU Abu Dhabi campus, which is located in Saadiyat Island in Abu Dhabi, which is the capital of the United Arab Emirates. And this is where we are.
Hi guys, so next to the microfluidics space, we have the cell culture room, and my colleague here will be taking you on a tour of our cell culture room.
Hi, I'm Pavitra Sukumar. Now I'll take you to the cell culture room, which is the space over here. And we have a tissue culture hood and other necessary equipments necessary for the cell culture, as well for the clinical samples which we work on.
Hi again, this is Ayub. So I spend a lot of my time with these friendly machines here, 3D printers, to develop probes such as this one. We call them microfluidic probes, and we use them for cancer research.
I'm Pavitra. Again, I spent some good amount of time in patterning filter papers for 3D cell culture cryopreservation.
Hi, this is Ana Saleh, and I'm working on microfluidic methods for single-cell transcriptomic analysis.
Hello again everyone. Now I will take you to our microscopy room.
Hi guys, welcome to our shared microscopy room, where we share with another lab, and we have several microscopes. And for us over there, we have a stereo microscope, and over here we have our inverted microscope, and finally we have our atomic force microscopy right here.
In our lab, we use atomic force microscopy to measure nanomechanical properties of circulating tumor cells.
Hi, it's Sam here again, and this is where I spend most of my time working on this multiphysics microfluidic probe for single cell analysis. And this is the setup where we have the electrical function generator, the XYZ micropositioner for the probe, the inverted microscope, and of course the microfluidic part.
Well, I hope you enjoyed the tour in our lab, and also we are lucky enough to have access to multiple core facility platforms here in NYU Abu Dhabi as well as in NYU New York. And we're lucky enough to have one of these core technology platforms just next to our lab, which is the clean room. And my group members will get you inside and show you some equipments on this clean room and how we find it very useful for our research in microfluidics and µTAS in general.
So let me show you around our core facility clean room and some of the machines that are available here. For example, we have a profilometer here, some microscopy, and we also have the nanoscribe. And chemical vapor deposition as well as reactive ion etching.
I'm excited to join Microdevices this year so that I get to meet a lot of leaders in the field.
Microdevices is important to me as a conference. I make sure that I go there every year because of exposure, collaboration, making new friends.
So I attend Microdevices every year because it's one of the leading conferences in the field, and Microdevices to me is a place where I can network with people and also learn off other leading researchers in the field.
Every year at µTAS, I get to meet my mentors, colleagues, and some future students and researchers. I can't wait until I meet you all in µTAS this year.